So very much thanks to the Mason family and to the members of AAAS for this great honor. I'm thrilled to be here to talk to you about my research today. And I'm a structural biochemist. And for those of you who may not think about what that means every day, I have some examples here of common household items where the structure is very important for the way the item works. So you can imagine, for example, this key, and it opens a lock. But not any key can work to open that lock. It must be a very specific shape in order to do its function. And this turns out to be a very common theme in biology, where the way that something is shaped is important to the way that it works. So my job as a structural biochemist is to look at the structure of things to understand how they work, and my great love since the time of graduate school has been that of viruses. And speaking of great female mentors, my PhD advisor, Carol Teschke, is here with us today, and she got me excited about viruses. Many of you in this audience have had an encounter with a virus at least once in your life. It's maybe not been so pleasant, but you may have had flu or the common cold, which is rhinovirus. You might be thinking about other viruses that you may have heard of. Could be herpes virus or something like a reovirus, or even we've heard it the last year, plaguing West Africa has been Ebola. So if you start to begin to think how many viruses are out there, you might think 10, 100, 1,000. It turns out there's a great diversity in viruses. And so to put this into perspective, a number that everyone here is probably familiar with is 1 million. In scientific terms, that's 10 to the 6th. Turns out there's about 10 to the 31st viruses in the biosphere. This is such a huge number, I don't even know the name that you would call it, but they are highly, highly diverse. And this presents a problem for biologists if we want to understand how viruses work. We couldn't possibly study all of them. So we do what scientists do, and we study model systems. So we use examples of some viruses that tell us a great deal about many other viruses. And as a structural biologist, for a long time, we have only been able to look at the very small and very simple ones because of technological challenges. So to give an example of some of the ones I've worked on throughout my career, this one is an example of Trichomonas vaginalis virus. It infects a parasite that causes a sexually transmitted disease. This is an example of reovirus, which causes great intestinal distress in young infants. This is an example of a virus that infects a bacteria, and this particular one infects a bacteria that causes dysentery. And this particular example infects also a bacteria that causes bacterial meningitis. And as you can see, the structures are very different. They're very beautiful and elegant. And the bottom two examples have a very striking appendage called the tail, and that's actually how they infect the cell. So by studying these structures, we can begin to understand how they attach to their host and transmit their genetic material. Now, as beautiful as these look and may be complicated to the untrained eye, these in fact are quite simple. So to get at this challenge, we have to devise new methodologies and new technologies in order to look at the highly complicated and large systems. And to do this, I'm using a system called cryo-electron microscopy. This is a picture of the microscope that I use, and that's a picture of my student Jason Schrad, who's in graduate school, preparing samples to put into this microscope. Now, these toys are a little bit rare in the US, and they're very expensive. And they're prohibitively expensive to operate if, as Allison and Kate alluded to, you want to start doing high-risk, high-reward research. So the award from the Mason family is going to allow me the opportunity to start looking at some challenging specimens by getting some time on these instruments to really start to look at the structures of complicated things. And the one that I'm interested in is one of the largest viruses in the world. This is a picture from that microscope. And to put it into perspective, just how large this is, these are examples of the viruses I showed you before, and this is how big the largest in the world is. And it looks pretty impressive on this screen, but I wanted to use an example in your day-to-day life that may really sink the message home. Most of you probably have used a ladder before. You may have cleaned out the leaves in your gutter or liberated a frisbee on the roof of your house. Typically, ladders of this sort extend between 15 and 20 feet. So if that's the size of the first class of viruses, the small and simple ones I told you about, the large virus that I just showed you would actually be a foot taller than the Washington Monument. It's huge. So on that note, I would like to say thank you again to the Mason family and AAAS for giving me this opportunity to start to study these large and challenging specimens. Thank you to my advisor who's here, and also Shannon Doyle, who is working at the NIH right now, who was actually getting her PhD at the time and was my TA who got me into biochemistry. My parents are also here and my other good friends are visiting. Thank you to all of my colleagues. And lastly, thank you to all of you in the audience for your kind patience and attention.